Hello, I welcome you back to the Keynote session number 2 with Dr. Emily Shacter from ThinkFDA LLC. Dr. Shacter regulated therapeutic proteins at the FDA for 18 years, last serving as the chief of the Laboratory of Biochemistry in CDER's Office of Biotechnology Products. Prior to leaving the FDA in 2012, Dr. Shacter supervised the review of regulatory submissions for a wide range of protein products. She contributed to the FDA policies and expectation for the manufacture, control, and analytical characterization of novel therapeutic proteins and biosimilars. Dr. Shacter received her PhD in biochemistry from Johns Hopkins University in '82 and carried out investigator-initiated laboratory research in cell regulation, chronic inflammation, protein oxidation, and cancer at the NIH and FDA for over 30 years. She now works as an independent consultant through her business, ThinkFDA LLC, providing strategic, scientific, and regulatory advice on FDA requirements for the development and approval of protein products spanning novel proteins, biosimilars, and breakthrough therapies. Dr. Shacter?
Hi, thank you, Harold. I should have written a shorter bio so I wouldn't have lost so much time for my talk. But good morning or afternoon, everybody, wherever you are. So what I'm going to talk with you about today is the important subject of how protein structure affects function. And what I'd like to be able to give you, in addition to providing thoughts about how you should be viewing this, but I'll also try to give you as much input as I can about how regulators think about this protein structure function relationship. And I'm sorry, I have a little bit something going on with my throat, so I apologize if I need to clear my throat or take a drink every once in a while. So here we go. So what we'll be talking about is I'll give an overview about protein structure and function, talk about how we evaluate protein variance and heterogeneity, and I'll give a little bit of a deeper dive into trisulfides today, and then give you a variety of case studies to see how the thinking is applied to the regulation and approval of protein products. Very quickly, just go through the basic hierarchy of protein structure, as you all know so well. Primary structure, secondary, tertiary, and quaternary all need to be evaluated as part of the evaluation of protein structure and function. And we need to bear in mind that proteins have multiple domains in them, so macromolecular domains that are both involved in controlling the protein itself, as well as controlling how, or regulating, determining how the protein works in the body. So we may have multifunctional domains that we have to look at, and this is true for many of our proteins. Now, proteins are generally heterogeneous mixtures of a population where we see a variety of variants in the protein structure. And I won't go through them bit by bit, but the point I want to make is that because proteins have many amino acids, many atoms, many elements to them, often they can tolerate certain variants, changes to the protein or amino acids, whatever, that have really no impact on protein function. Our challenge really as protein developers and regulators is to determine which of these attributes matter, which one impact function, and which ones don't. Contrast this to a small molecule drug, so in this case, a couple of hormones, where you have very few atoms and molecular linkages, and changing just a few of them has tremendous impact on the biology of that chemical. So for example, comparing what outcomes that you have from testosterone versus estrogen. If we move into a small protein such as insulin with 50-odd amino acids, changing just 3 of those amino acids can have tremendous impact on, for example, the solubility of the protein in the face of certain pHs, and consequently how the product behaves in the body and how it is distributed into the body, which greatly impact the half-life of the protein in the body. So a small protein changing just 3 amino acids. We can look at another very different kind of protein, a growth factor, so erythropoietin, which is relatively small in its protein, 165 amino acids, but is heavily glycosylated, such that roughly 40% of the molecule is glycoforms. And these glycoforms greatly impact the interaction of the protein with the receptor and also the biodistribution of the protein in the body. So now we're looking at a multidimensional aspect of the protein, looking at both the glycoforms and the protein structure that's responsible for binding to the receptor, which is responsible for its biological activity. Then move into a much larger protein, a monoclonal antibody that has, in excess of 1,400 amino acids. And here we know an awful lot about what each amino acid in this protein may contribute to the function of a variety of different antibodies. By virtue of the large size and many of the amino acids really don't contribute to function, an antibody can tolerate a lot of different variations in various aspects of the protein and we still see no impact on function. So here we're looking at a spectrum of the range of protein size and complexity that helps us understand, again, ask the question, which variants, which changes to a protein, which structural elements are responsible for activity and biological function in the body and which aren't. Now, I won't go through this slide in detail, but we have many tools available to us. Those of you in the audience probably know all of these even better than I do. But we have many tools available to us to evaluate the many different elements of protein structure, whether it's the folding, whether it's modifications to the amino acids, whether it's elements that we've added onto the protein to change the half-life, protein-protein interactions, et cetera. And so we use all of these analytical tools to try to get a handle on the relationship between protein structure and function. Now, in terms of what we all recognize is that analytical product characterization is critical to the clinical development of a protein product. And it's absolutely essential that you know your product. And what this means is that you need to identify the critical quality attributes of the protein, or those structural attributes that are critical for function and the clinical profile of the product. And typically you'll be doing this in research and development, and then develop assays to help you evaluate the relationship between any particular protein structural element and the function of the protein. So what we need to know in general is, what is the desired protein? How does it work? So what are the mechanisms of action that are responsible for efficacy? How well does it work? So what is the potency, the specific activity, for example, of the protein, and what is it targeting in the body that determines how it works, which is the pharmacodynamics. How and for how long does it get around the body? So the pharmacokinetics of the protein and the bioavailability to the target sites in the body. Where does it go in the body? What is the biodistribution? What are some off-target effects that may or may not have to do with the actual desired molecular function of the molecule? And this determines safety and sometimes immunogenicity. And then how much of the protein is enough? So we think about the therapeutic index of a protein, where we have enough to get the efficacy that we want, but we don't want to tip it over too much into also then adding safety concerns for the molecule. So where do we find the risk-benefit balance of how much protein is enough to have a good clinical profile? The main elements of analytical protein characterization then are, why does the product work in the body? So again, what are these critical function? What is tissue distribution of the molecule, which is highly dependent upon its structure and also its stability in vivo, how it gets broken down, what receptors it's binding to, how it's being eliminated, what aspects can interfere with expected with some case examples that I'll give. So if we have poorly understood variability, we can have unexpected clinical outcomes. If we have an inactive or less active protein variant or impurity, this can impact clinical activity. If the bioavailability is somehow impaired by some change in the molecule, for example, glycoforms, that can impact its getting around the body. And then immunogenicity. So when antibodies are developed against a protein product, they can change both the PK and the biological activity of the protein through neutralizing activity. What else is in there? So what impurities do we have in our protein product that we need to understand? And then how does the product change over time? So is it stable under the recommended store conditions? And if not, what aspects of the protein are changing? Oops, and there we go. So now I want to go into some specifics of various different kinds of protein modifications and talk about how we need to evaluate them, starting with clipping or proteolysis of a protein. And there's many questions that we need to answer about this very interesting and very complex topic. First and foremost, where in the molecule does the clipping occur? If you see clipping occurring, for example, in a capillary electrophoresis system, does the clipping occur in an active site of the molecule, or is it in a non-functional framework portion of the molecule? What causes the clipping? Is it due to a protein impurity that may have been present, for example, in the host cell proteins that co-purified with the product? Or is it due to some chemical cleavage and instability of the molecule? Does the cleaved molecule remain intact after cleavage? So, for example, through disulfide bonds or hydrophobic interactions that might keep the clipped portion of the molecule together, which then may not result in any change in function. Does the clipping affect bioactivity? And is the clipped molecule more immunogenic, for example? Moving on to another kind of protein modification, which has less apparent impact, but still may, on protein function. So we look at our aspartate isomerization, which is very common, and it could have an impact on bioactivity depending on where in the molecule the aspartate isomerization occurs. Again, if it's in an active site or a complementarity determining region, or if it causes a conformational change in the protein. It could cause an impact on the PK of the molecule, and I'm going to show you a reference for that. It's not above, it's below. And aspartate isomerization could, in theory, impact immunogenicity. There's at least one report of a case where there's an isoaspartylation and a histone that was found in patients with lupus that resulted in the formation of antibodies to the protein. Because impact, even though aspartate isomerization may usually not matter for protein function, because it has been seen and can be seen, regulators don't assume that it's not important. And therefore, when you see it or if you see a change in your molecule, you need to justify the change that you're seeing. Another common form of protein modification is oxidation. And here we need to understand which amino acids are being oxidized. A variety of amino acids in the protein can be oxidized. The one we see the most is methionine because it's most highly oxidizable. But depending on the level of oxidative stress, you can also see other amino acids being oxidized. Where in the molecule is the oxidation occurring? So again, is it happening in a binding site or in a framework region of the protein? We've seen that oxidation of an antibody in the binding regions of the antibody can cause a loss of binding activity. And similarly, it has been seen that when we have oxidation of methionine residues in the FC portion of an antibody, it can impact FcRn binding, which is responsible for the half-life of the molecule in the body. Turns out that you need an awful high level of oxidation on both of the heavy chains in order for this to occur. So typically small changes in methionine oxidation in the FC portion of an antibody don't matter, but nonetheless, it has to be paid attention to. And then if you have oxidation, for example, in N-terminal methionine, it's highly unlikely to be important. That methionine is kind of waving around in the wind on the protein molecule. what type of molecule is being oxidized. So there's a big difference, for example, if you have oxidation in an antibody, where it's in the active site or in the framework region, whether you're in an active site of an enzyme molecule or whether you're in a small molecule inhibitor, such as a protein, which can have great impact on activity. And then finally, we need to understand how oxidation might impact immunogenicity. Typically, the oxidized amino acids themselves are not immunogenic. So methionine sulfoxide, for example, it's very hard to get antibodies to that amino acid modification. However, oxidation can cause changes in folding of the protein. And this is more likely where increased immunogenicity may be coming into effect. In general, protein modifications impact different proteins differently. And I've been trying to say this already throughout the talk. But if we take oxidation as an example, you can have oxidation of a single methionine residue in alpha-1 antitrypsin, which completely knocks out its elastase inhibitory activity, which is one of the underlying causes for emphysema. So a single methionine in anti-1 alpha trypsin compared to the oxidation of multiple methionines and some other proteins, which actually has no impact on function at all. So understanding where and how much any of these modifications happen is key to understanding the impact on function. One more change that I want to talk about briefly is the Fab-arm exchange in IgG4 antibodies. So because of the unique structure of the hinge region, an IgG4 molecule can dissociate into half antibodies. And if you have other IgG4s that are also dissociating into half antibodies, and these can come then back together, you can have several outcomes from the mixing of the half antibody from one IgG4 to that of another. And this is called the Fab-arm exchange. You can have many different outcomes. For example, you can have a loss of activity because you've just lost half of the active portion of your molecule. You can have gain of a bifunctional activity. You can have different targeting of the protein in the body because now you have a different molecule being targeted in the body. And then you can consequently have unknown clinical outcomes. How can we prevent Fab-arm exchange? Well, mutation of a single serine 228, to a proline, which you can see here on the bottom of this slide, actually prevents the IgG4 chain switching. And this is really beautiful work that was done quite a long time ago. So many sponsors who are developing IgG4 molecules actually introduce this modification so that they don't have to worry about all of these potential sequelae. The FDA still wants to understand, the FDA, which is the regulatory organization that I know the best, still wants to understand if you're developing a molecule, whether you are seeing Fab-arm exchange, even if you have the amino acid change from the serine to proline. But I think over time, the regulators will stop looking at that as much as they have been to date. Now I want to take a deeper dive into trisulfides. And the reason I want to talk about them is because we don't talk about them very much. Yet trisulfides are a common modification that we find in proteins. A trisulfide occurs when you have the insertion of an additional sulfur molecule, sulfur atom, moiety into a disulfide, leading to the formation of a trisulfide. This is a chemical reaction, but it occurs endogenously in the body through normal processes where the body, through the enzymatic breakdown of cysteine and homocysteine, results in the formation of hydrogen sulfide, which then, under appropriate conditions, can insert that sulfur into a disulfide bond to form the trisulfide. Trisulfide bonds tend to be stable, but they can be reduced back to the disulfide through mild reducing agents. If you look at an antibody, we actually see that there is some site specificity to where trisulfides are formed. So they're formed most commonly in the light chain to heavy chain interactions, which you can see here, or also in the heavy chain linkages. They're very rarely seen in the And about 10 years ago, a group at Biogen did some studies to see, all right, well, how much trisulfides do we have in the research antibodies that are being developed? And Gu et al. Found that you can actually have quite high levels of trisulfides in a protein, as high as 39% from this study. So they are formed. They formed naturally, and they consequently need to be followed and understood. Trisulfides have, in fact, been evaluated in a variety of therapeutic proteins, including EPO-alpha, which I'll talk about a little bit more in a case study. They've been found in growth hormone and a molecule called truncated IL-6 and the other proteins that you see here. So what do we know about how trisulfides impact function? Well, when it's been looked at in monoclonal antibodies, there's actually been little or no impact seen on either ligand binding or bioactivity. And the same is true when the impact of trisulfides has been looked at on human growth hormone. But two other molecules, oxytocin and IL-6, have actually seen that when trisulfides are formed, they can result in a decrease in receptor binding and potency. So effects have been seen on bioactivity. Therefore, they have to be studied. So in one particular case study, excuse me, that I'd like to give you that helps us understand both how the FDA and a sponsor evaluate the impact of trisulfides came in the development of a biosimilar to erythropoietin by Hospira before they were acquired by Pfizer. And what Hospira found was that their Epogen their erythropoietin biosimilar had about 4.5% trisulfides in a certain cysteine disulfide bond that was not found at all in the reference product. And so everybody had to ask, is this protein suitable for being developed and approved as a biosimilar when there's a structural change that is not seen at all in the reference product? In the end, the FDA concluded that, in fact, it was acceptable to approve this product as a biosimilar. And the thinking that went into this was as follows. The conclusion being that there was not expected to have clinical impact. So earlier lots of epoietin Hospira had even higher levels of trisulfides, greater than 10%, but they had the same in vitro and in vivo specific activity. So there's good assays for EPO activity, and there was no impact of even higher levels of trisulfides compared to those found in the proposed commercial product. There was similar receptor binding with and without the trisulfides. The literature suggested that trisulfide species are also formed in vivo. I don't find that the most convincing argument for this kind of thing, but that was one of the arguments to accept it. And finally, there were studies from the article that I showed you just a moment ago that showed that trisulfides are actually converted back to disulfides within about 24 hours of being injected into the body. So for all of these reasons, the FDA concluded that there was no potential clinical impact from this structural change, their difference in a biosimilar, and it was approved as a biosimilar to Epogen. I wanted to add just a couple more of my own perspectives because I did do a deep dive into what we need to understand about trisulfides, and we have to look at two other things, and those are the potential impact on PK and immunogenicity. The fact is there's no publicly available data to demonstrate or refute a potential impact on PK, although in the studies with Hospira's EPO, no effect was seen. And I think from my survey of the literature, the potential impact of trisulfides at least some lower reasonable level is unlikely unless there's some change to the higher order structure of a molecule that can impact how the body sees it and clears it. Now, similarly, for immunogenicity, there's no publicly available data to demonstrate or refute an impact on immunogenicity. However, the trisulfide bond itself is not immunogenic. So if there is going to be immunogenic impact, it's likely again to come from a change in the conformation of the molecule. So now I want to move into some case studies to try to show you how both sponsors and the FDA think about how when we have changes to a molecule or differences, for example, if you're doing comparability or biosimilarity studies, how we understand the relationship between structure and function and how this leads to regulatory conclusions. So the first case study is Myozyme. This is a very old case study, but it does allow us to make a couple of important points. So Myozyme is an enzyme that it's a replacement enzyme used for the treatment of a glycogen storage disease called Pompe's disease. And it comes from the deficiency of the lysosomal enzyme alpha-glucosidase. The enzyme has end-linked carbohydrates, and important among them is mannose-6-phosphate that is responsible for controlling the uptake and the intracellular trafficking of the protein into the lysosomes and the target cells, which are muscle cells in this case. And this happens to the mannose-6-phosphate receptor. The less mannose-6-phosphate you have in the molecule, the less uptake you have into the lysosomes, and consequently, the less efficacy you have. So what happened when Genzyme was developing the protein for approval, they made a pre-licensed manufacturing scale-up, and the FDA reviewers noticed that there was a potentially significant change in the levels of mannose bisphosphate in the scaled-up molecule, where there was actually a lower level. A small difference, we're talking about several percent here, but because this was a known critical attribute responsible for the uptake of the protein into the target cells, then it was evaluated very deeply. And both the CMC reviewers and the clinical reviewers decided that this post-change product was not comparable because it had lower efficacy in patients with infantile Pompe disease, and attributed to this small but important molecular change. So actually the sponsor had to submit a new BLA in order to get approval of the scaled up product. The product became Lumizyme, and it was used, the indication is for late onset Pompe's disease. But what is the point of all this? The point is that seemingly small differences in a protein can be clinically meaningful. Do small differences always matter? No, and in many cases they don't. But we need to understand, we need to do the structure function studies to know which small or large differences matter and which ones don't. Now, the next couple of case studies that I want to give you come from biosimilars, studies with biosimilars. I already talked about the Epogen case with the Trisulfides, and now I'm going to go into two more. The first of which is Enbrel, for which the INN is Etanercept. And when Sandoz was developing this product for approval, they noticed a relationship between correct and incorrect disulfide bonding and bioactivity of the molecule that greatly impacted their development of the product. And what they found was that when they analyzed the reference product Enbrel, over time, over the years of development, they found that the mean activity, TNF alpha binding and subsequent activities of the molecule, apoptosis, the mean activity actually appeared to be decreasing over years of the distribution, of the commercial distribution of Enbrel, where for their product, because they weren't having whatever issue there was in the Enbrel, they had consistent bioactivity that, unfortunately for them, had been targeted to the earlier mean and not the later mean. What they found was that the apparent drop in bioactivity, in the mean bioactivity of Enbrel, resulted from an incorrectly disulfide bonded variant, which when subjected to, let's say, physiological redox conditions, could be converted to the correct disulfide bond variant. And so when they looked at, for example, their own product that had various levels of the trisulfide, they used various manufacturing means to try to identify these molecules. They could see a relationship between those samples that had higher levels of the trisulfides and lower activity versus those samples that had lower levels of the incorrectly linked disulfide and higher activity. So they had correlative data to show a relationship between incorrect disulfide bonding and activity, but they also took embryo lots and subjected them to this redox system and found that they could restore the activity from the lower to the higher activity by converting the protein to the correct disulfide bonded structure. So they had both correlative and experimental data to confirm the relationship. And this allowed them to actually get ultimate approval of their product because they had an understanding of the relationship between the disulfides and biological function and potential clinical activity, which would be the FDA's main concern here. Okay, so before we move into our last case study, I want to introduce the subject by saying, what are our major CMC goals? Well, we want to manufacture a consistent product with a well-understood safety and efficacy profile. That's the bottom line, right? And we don't want to have any surprises to the change in the safety and efficacy profile that result from manufacturing changes that we may make. We don't want to have any changes in the clinical profile or any interruptions in release and distribution of the products. So this brings me to our last case study, which comes from Samsung Bioepis' work in their development of a Trastuzumab biosimilar, so biosimilar to Herceptin. And what they found were certain glycoform and activity changes that were very important for both understanding marketed Herceptin as well as getting approval of their product. Very interesting case story here. Trastuzumab, or marketed product for the Genentech product is Herceptin, is a monoclonal antibody. The Fab region is responsible, of course, for antigen binding. And just binding of the Fab region to the target, which is HER2 on the surface, in this case of breast cancer cells, can inhibit proliferation of those cancer cells. The FC portion of the molecule, which is also glycosylated, is responsible for the effector functions of trastuzumab, which are very important for the clinical function of the protein. And these include antibody-dependent cellular cytotoxicity, or ADCC, and also binding to the FC gamma receptor of this portion of the molecule, which leads to its bioactivity when it binds to effector cells. There's a well-known relationship between non-fucosylation of molecules that have effector function, such as trastuzumab, and ADCC activity, where the more afucosylated product you have, the higher the ADCC activity that you have. And this slide happens to be taken from some data from Sandoz, from a public talk, obviously. And so what Samsung found when they were, again, looking at Herceptin's, commercial Herceptin samples over time, they found that, let's start with the ADCC activity down here. They found that at some point in time, all of the samples that they collected from the shelf, from the marketed product, actually had a lower ADCC activity. That then, looking further over time, it actually seemed to shift back up again. This decrease in ADCC activity over time correlated with a drop in afucosylation and high mannose. Those two tend to go together, and then a shift back up over a second period of time back to the levels that were seen previously. And again, this correlated reasonably well with looking at FC gamma R3A binding activity, which is responsible for binding of the drug to the effector cells that are responsible for causing ADCC-mediated killing of the target cells, which again, in this case, is breast cancer cells. What the Samsung researchers found in addition, and very importantly, was that this shift in activity, ADCC activity of Herceptin, correlated with lower clinical efficacy of the product, so a lower event-free survival of patients. This is very important because if, for example, the sponsor was on, and I don't know what was being studied for release of the product, but if anyone were just looking at HER2 binding or looking at the anti-proliferative activity of the protein, which derives only from the Fab portion of the molecule, you would never know that anything had changed in the product. It was only through a deeper analysis of the FC portion of the molecule and the FC-mediated effector activity that it was seen that there was a change in the product, a drift in the product over time that had an impact on clinical activity. This is exactly what we do not want to happen. We don't want to have unexpected changes in clinical safety or efficacy profile. This brings us to the important point that how do we know the relationship between structure and function? Well, we study function. And we do this through a variety of methods, including potency assays. So relevant functional assays are, to date, the best available predictors that we have for determining what the clinical activity and efficacy profile is going to be. So what we need to understand is, when do protein modifications matter? And we do this through the use of the bioassays that we have, and quite a variety of them often need to be applied to many molecules, especially when there are multiple domains in the molecule that control the clinical activity of the product. We need to evaluate the binding and bioactivity of variants through CQA analysis. So what are the critical quality attributes of a protein? How do we know? We do these kinds of studies. So doing the CQA analysis, the assessment of which protein attributes matter, is actually essential for product control and process control. If you don't know what matters, what elements of manufacture can impact the important structural elements of a protein, you can't control your process or your product. Often this requires, and some changes to a molecule can be in the low percentage range, but as we've already seen, a change in a low percentage range can be important. Then often we have to isolate and enrich the modification that we're trying to study in order to determine whether there's an impact on potency. Because if you look at a 3% change, let's say, amongst the whole population of molecules, you're not going to see an impact on potency. We also have differences, for example, in using binding assay versus cell-based bioassays for mAbs or for other kinds of proteins. And some of the considerations that we look at for looking at binding and cell-based activity can be a little bit different, for example, when we're looking at antibody drug conjugates, which are delivering a toxin to the target cell. When we're looking at enzymatic activity, we have to look both at the activity of the protein and those elements that are responsible for uptake of the protein, for example, for a replacement enzyme like we talked about with Myozyme. And then for growth factors, we look at receptor binding and also the cellular response to that cellular binding, whether it's some signaling event, proliferation, apoptosis, whatever the functional outcome is from that binding. So last year at the WCBP meeting, and I'm almost done, I think I'm within time here, we had a roundtable where we tried to talk about structure-function relationships, and we came up with a list of additional strategies that you can use to try to help you understand whether a change in structure, for example, for a comparability or similarity exercise, is actually important. And first and foremost, you need to determine whether the structural difference that you're seeing is an actual difference or whether this is an artifact due to some element of the method being used, maybe an artifact due to the reagents being used. So make sure a difference that you see is true, oftentimes using orthogonal methods to try to understand. You could use pharmacodynamic markers to determine if you have a change in your product, whether there's an impact on the clinical profile. And this can sometimes give you an earlier read on whether there's been an important change to the molecule, short of doing an actual clinical efficacy study. You can use genetically modified cells to evaluate the role of some attribute, like a glycoform on PK or on function. So, for example, engineering the cells to remove the moiety or the structural element that you're studying in order to see whether there's an impact. Force degradation studies are very important for trying to help us tease out whether a structural change matters. We may not see a change, the impact of a change in the molecule when it's stored under its recommended conditions, but you can tease out an underlying important change, which could then come out under circumstances such as physiologically that can actually impact the product. So we often do force degradation studies to help us both identify critical quality attributes and how they might impact function. If you have made, for example, a change in manufacture, you want to keep retains of your clinical materials because as you go through the commercial life of a product, you always need to link back to the clinically, the material that was studied clinically in order to make sure you're not going to have a change in clinical function that can otherwise only be determined through a clinical study. And then finally, if you have the resources, you could do some high-resolution pharmacokinetic studies to see, for example, how different glycoforms that you may be looking at impact the PK of the molecule. You can actually look at different glycoforms and how they are cleared differently from the body. So finally, I want to ask, you may be asking the question, well, this is hard work. Are we sure we have time and money to do all of these very important studies? And what I would ask in general of all of you is, can you afford not to do this? You don't want to have any of the surprises or hindrances to development and approval of your products by not having done this kind of a deep dive into your protein structure function relationships. And finally, I just want to thank my former coworkers at the FDA. And now I learn a lot from working with my clients. And thank you all for your attention.
Thank you, Emily.
The camera. Ah, there we go. Okay. Okay. Yep. Oh, who's talking? Harold's talking?
Shall I start?
All right.
Okay, then I'd like to thank you for this very interesting talk, Emily. We now have time for a question and answer session. The questions have been piling up.
Okay, I'll start with a question, Emily. Have we reached a stage or reached a position where higher order structure should now be a release assay, or is it still okay for characterization only?
Yes. So when we're talking about higher order structure, so for example, looking at circular dichroism or FTIR or other studies like that, I think that the truth is that these are relatively crude tools to be able to, they're informative, they're very important. But no, they are not typically part of release. I don't think that they're going to be part of release in a long time. But I will say that there are some chromatographic methods that we use that actually do tell us something about higher order structure. So for example, when you have hydrophobic interaction chromatography or reverse phase HPLC, that can be sensitive to the folding of a molecule and the exposure of, for example, formerly buried hydrophobic regions. They actually do tell us something about structure function, about higher order structure. So actually, we do measure it in some way. And of course, what most people use are the bioassays, because those are the ultimate arbiters of whether we have the correct higher order structure of a molecule.
Okay.
So then I would just hop up to the next question from Nadine Ritter. Can you comment on two opposing perspectives that I frequently hear regarding the orthogonal QC toolkit for biological therapeutics? One, if the product is being controlled with a sensitive specific bioassay, why do we need to use physicochemical methods to assess elements of structure? Versus two, if the product is being controlled with sensitive specific physicochemical methods, why do we need to use a bioassay to confirm potency?
Yeah. It's a great and age-old question. When we talk about having commercial protein products, which are really relatively complex compared to those kinds of molecules that have typically only been understood through the chemical or structural studies, we actually want to have a depth of understanding to know that we're actually not having any change in the product. So actually, a couple of the examples that I gave show you how you can have a small change in the protein that actually has a large impact on activity. In fact, the 3 case examples that I gave really relate to that. A several percentage change in Myozyme, which if you see a 3% change in mannose bisphosphate, if you're only looking at physicochemical methods, how are you going to know if that matters? If you have only a 1 or 2% change in the levels of non-fucosylated protein in a protein that has ADCC effector function, how would you know that matters? And in some antibodies, for example, it may matter more than others. So we always want to have a depth of understanding of what are these products that we are putting into people and on which we're relying that this year's batch and next year's batch and the batch from 10 years from now are going to be the same. So I would say that some of this is about the depth of analysis. I would also say, in my own opinion, that there's nothing like a bioassay, because in the end of the day, you can boil a molecule, you can have the same, maybe let's say peptide map, and maybe you can even have a couple of other chromatographic methods show no difference, but you ain't going to have a product that works in the body. So I think we need both. And I think that will continue to be true for the foreseeable future.
Thank you. Next question. It may be quite difficult to study each protein modification individually. Given the increasing availability of in silico modeling tools, do you think that it would be acceptable to present outcomes of modeling as a way of showing what we know, the molecule and the potential impact of its modifications on function?
Well, gee, I wish I could ask the person asking the question a question. But in this day and age, I skipped pretty quickly over the slide where I showed all the analytics that we have available to study proteins. But in 2020, we have so many tools available to us that, and one more point before I finish that sentence, with the advent of biosimilars, where we have really extensive characterization of proteins far beyond what we used to do 10, 15, 20 years ago, I'm trying to think of a molecular attribute for which we don't have an analytical tool. So we can use the in silico models to try to help us understand and predict structure-function relationships. And this can be done in very sophisticated ways. But I can't think of a molecular attribute that we actually don't have a tool to study. And I wish if the person asking the question can give me an example, then we can come back to this question.
Okay. I'll leave it open to the questioner to add a comment.
Or we can move on and come back to it if we're going to run out of time.
Yeah.
Okay. Well, then the next question is, how representative, reliable are in vitro assays, including cellular assays, for the prediction of clinical efficacy, or would that be a case-by-case study?
Yeah. Yes, it is truly case by case, but I think the first point that needs to be made is that when you develop a protein product, you are developing that product for a particular disease for a reason. And the reason is that because somewhere along the line, somebody has understood that there is a biological activity to the protein. Take one of the first molecules that I worked on when I was at the agency, a thrombolytic, so TPA. So it was understood that this molecule has the ability to lyse clots that are formed to try to reverse the effects of heart attack and stroke. And it was understood that the protein has multiple ways of being regulated. And there were assays developed to be able to look at all the various regulatory elements of the protein. So the point is that we don't just make a protein and then decide we're going to put it in the body and see what it will do. Proteins have activities for which we think they may have a clinical function. So that is why we use bioassays, and that's why they are the bottom line for trying to predict clinical activity. That said, there are some molecules, for example, that are being developed that they may hit so many different organs or tissues in the body that we don't exactly know how they're going to work. We have some theory, but we're not sure. And so therefore, we don't know which bioassay to try to develop to try to predict how they may work in a different clinical setting. Indication. And for example, oftentimes there's proteins where I have a function, but I'm looking for a clinical indication for which it might work. And then it is very difficult to develop a relevant bioassay. But you do the best that you can because you're not developing this protein just out of thin air. You have a rationale for developing it, and that almost always comes from its biological activity. Therefore, you can develop an assay for its biological activity, for your proposed mechanism of action for clinical activity.
Okay, thank you. Next question. The presence of trisulfide seems to not affect bioactivity, for IgGs I'm talking. Is there anything known whether formation presence of trisulfides could be related to the appearance of higher levels of fragmentation, e.g. LC and HHL fragmentation with the trisulfide on the LC-HC linkage? Is there anything known? Do others have experience of this? We think we see a relationship in our products, even at low levels of trisulfide.
Yes, well, that's a really interesting question. And so in my deep dive into trying to look at how do trisulfides matter and how do we understand them? And I did that specifically for this talk because it's something that's not talked about a lot. I didn't see anything on that point, but absence of evidence is not evidence of absence. So I think it would have to, so since the trisulfide bond itself is stable, and since the trisulfide bond is also naturally reduced back to a disulfide through the redox systems that we have in our body, our glutathione systems and et cetera, redox conditions that we have in our bodies, then it wouldn't be my first guess. But if you have a structural change in the molecule that sort of makes a linkage less stable somewhere, then you could have a chemical clipping of the molecule. And so what I would suggest is that you definitely work this up. So I have no evidence of this. I don't know anything about it. That's the bottom line answer. But work it up and then get it out there because this would be really important to know.
Okay, jumping in on trisulfides. Is the formation of trisulfides in a molecule accompanied by steric stress, or does a trisulfide only occur in regions where there's enough conformational flexibility?
Well, another really good question. And I did not see anything in the literature on that, but it is interesting that it occurs in interchain linkages and not intra-chain linkages. And you might think that those intra-chain linkages simply can't tolerate it because of their lack of flexibility, whereas in interchain linkages, you're expanding the linkage by a sulfur, by an atom. And so you can imagine that you have more sensitivity there. But I didn't see anything to show, for example, that this can actually result in the separation of the one chain from the other. So it's a really good question. I didn't see anything on it, but I think because these are mostly found in interchain linkages, I think the molecule can tolerate it by that one atom shift. And a good question to study. Very good question to study.
Okay. Next question. In the formation of trisulfates, in a molecule accompanied by steric stress, or does a trisulfide only occur in regions where there is enough conformational flexibility?
That was the last
Yeah,
question.
so that's what we just talked about.
Right. So sorry.
Yeah.
I'm trying to find the top ranked ones. Okay.
Yeah, I have the same problem here. So perhaps just jump in. So we heard a lot yesterday about emerging technique of NMR for looking at higher order structure and the cost mentioned. Interesting to hear the comment about affordability from you. NMR is certainly not a crude technique and offers insightful picture into what is happening at the atomic level. What would be your view to look at NMR data in release testing?
Yeah, okay, so not release testing. I'll answer that question quickly. However, it is not release testing. It's not practical, but it is such a powerful tool and has been used very effectively to be able to, for example, show both yourselves and the FDA that, for example, when you're developing a biosimilar, when you can look at the relationship of every atom to every other atom and show that my molecule is the same as somebody else's molecule, or at least highly similar, oh, that's a very powerful tool. So both 1D and 2D NMR have been very useful for that. But and one could say, well, you could have a CRO do it for your release testing, but it's not going to happen. And most companies don't even have an NMR equipment, so it's not going to happen. Hey, we're just talking about getting mass spec into the QC environment. So maybe in 50 years, when they make a little tabletop NMR, then maybe we'll talk about it again.
Be realistic. Ask for the impossible.
Exactly, but it's not going to happen now.
Okay. You can have a discussion with Eve over that. All right. Regarding the Myozyme, we haven't done this one, I hope. Regarding the Myozyme case study, you mentioned small difference in the levels of MAN6P did matter. Are we talking values less than 1%? This means the acceptance levels for N-glycans would depend on the specific glycoform.
Yes. So I don't think I'm allowed to know the answer to that for you. I can say that it was in the low percentage levels. I don't think it was less than 1%, but I don't think that information is publicly available. But I think what we need to, but for example, let's take a different modification, the afucosylation that we've talked about. A 1% change in afucose actually does impact bioactivity. So when you're looking at low percentage differences in molecules, this is where my comment about where and how much in which molecule, where does it happen, how much does it happen, and in which molecule. And you have to do the isolation and characterization studies to be able to determine whether in this molecule a very low percentage change matters. I don't think I've seen a glycoform change of less than 1% that would have a dramatic or a significant clinically meaningful effect on protein function. But there are some that are close, that are in the low percentages that can matter. So I'm not exactly answering your question, but we always have this question, is there a limit below which I don't need to study the potential functional impact of a molecular attribute? And there is no hard and fast rule, and it's partly why I gave you examples of impacts of low percentage. But honestly, when you're looking at something in the 0.5% range, I don't think I've seen that matter. You use your vast experience to tell you, but it's really through isolation and characterization. So enriching whatever attribute you're talking about and looking for potential impact versus trying to study it in the larger population molecules in the product, that you'll get an answer.
Well, okay, we have several questions that go on in this strain, are in this line, and in principle, I would just ask the question as well, is typically you only have a handful of clinical trials. how do you tease out which 3-4 parameters are really worth looking at? Because, I mean, you can look at a dozen different things in your batches that might be slightly variable. So again, 1% change is, yeah, but obviously you have to look at it. How do you tease that out, which one to look at?
Yeah, and it's so important that you do it. So this is really where your CQA, your assessment of critical quality attributes comes from. And using your, for example, forced degradation studies, this really helps you figure out if you stress a molecule and then you see a change in either binding or activity, enzymatic activity, receptor binding, whatever, through that stress, then you know, so you look at all the elements of the molecule that may have changed in that stress. And then you do enough studies to be able to figure out, oh, it was this amino acid, this series of amino acids, or it was this structural feature, this disulfide bond that was responsible for my loss of activity in response to that stress. Whereas when I look at these other elements of the protein, I can modify them to 20, 30, 50%, and I don't see any change. So using the forced degradation studies helps you figure out which elements of the protein are responsible for function. So that's one really good way to do it. The stress studies modify portions of the molecule and see if they matter. And again, we have to do this in order to be able to control the process, because if you want to work at a variety of pHs, or you have, whatever conditions you're using to elute the protein from a column, and you're changing something that you don't know you're changing because you're not looking at it. And then you can have a change of activity. So this is the job of biotechnology in 2020. And, in 2020, we have a lot of tools available, and it is incumbent upon us to apply them. I'm not sure if I answered your question, though. Did I, or no?
Yeah, yes and no. Yeah. So I've been asked to slowly start wrapping up. Okay. So we can do that sometime offline. But okay. Again, thank you very much for this very interesting talk. And the number of questions we have left open, I'm afraid, we'll have to try and answer offline. So thank you all for participating. And I'm finishing up for this, and now we have the exhibitor showcase and posters.
Okay, well, thanks everybody for your time and attention. And it's been a pleasure to be able to do this, even though I do wish I was in Lisbon.
We all do.
Yeah.
Thanks, Emily.
All right.
Thank you.